Okay, so this chapter it's about specific details of prokaryotes. So we're going to see their classification, their subclassification, their characteristics in terms of where they are found, what family members do they form, and how can you identify them. So prokaryotes, we already know that based on gram staining, can be subclassified into gram-positive and gram-negative bacteria. But we have these specific groups of gram-positives and gram-negatives that they fall into different families based on ribosomal RNA signature. And bacteria then has this domain that is gram-negative, and then we have phyla that you can see here in this left column, and then classes, and then we have a specific nodes. So we're going to see the great family of the gram-negatives, which are the proteobacteria. This is the largest family members of bacteria that we know. And then, again, we're going to see what do they do. So we're going to see the first member of the proteobacteria in gram-negatives, which is the alpha proteobacteria. So we have gram-negatives in the pseudomonodota that are chemoheterotrophic. If you remember, chemoheterotrophs are those ones that obtain carbon source out of organic molecules, like proteins, DNA, lipids, etc. So this is the largest taxonomic group, and then can be subdivided into alpha, beta, gamma, delta, and Campylobacteriota. So alpha proteobacteria. This is metabolically diverse, so you can use many nutrients, and they can live in an environment where you have low levels of nutrients. This alpha proteobacteria, it is characterized by having stalks or buds emerging from their cells, which we know as prostacea. Members of alpha proteobacteria, Peligabacter. This one, it is found in the oceans, is the most abundant bacteria in the ocean. It is very small, and again, just like any member of the alpha proteobacteria, can live in low environmental, no nutrient environmental conditions. This bacteria plays a role in the cycle of carbon to recycle carbon sources. Another member of alpha proteobacteria, it is Azospirillum. This bacteria grows in soil and uses nutrients that are excreted by the plants, and in some cases can form associations with roots of many plants, like tropical grasses and sugarcane, and it fixes nitrogen. So it lives in symbiosis with plants. Acetobacteriaceae, we have two members, Acetobacter and Gluconobacter. So these bacteria are used in the food industry or so to produce ethanol and acetic acid. Members of alpha proteobacteria that are medically important, we have Rickettsias. Rickettsias are obligate intracellular parasites, so they need to live inside the cells. That's why they are obligate intracellular parasites. We have three important members that causes spotted fevers. We have Rickettsia prowazekii that causes epidemic typhus, and we have Rickettsia typhi that causes endemic murine typhus and we have Rickettsia rickettsii that causes Rocky Mountain spotted fever. These ones are transmitted by insect bites or tick bites. Ehrlichia causes ehrlichiosis, is transmitted by ticks. So these spotted fevers are manifested by basically having little spots in your skin that looks like reddishness. That's why it's called spotted fevers. Typically, these bacteria, especially Ehrlichia, they live in the endothelial cells, in the blood vessels. So endothelial cells is basically the most inner layer of our blood vessels, and they burst these cells, and then you have bleeding. So it's like a microbleeding in your skin. That's how you have these spotted fevers. And this is how these rickettsias look like. So this is a transmission electron micrograph. So it's like a rod, and then they have this slime layer. It looks like a capsule, but it's a little bit more disorganized. So this is Rickettsia. In order to grow them in the lab, you require cells because these are intracellular parasites. So here we have a fibroblast, or well, a chicken embryo cell. This is the cytoplasm. It looks like a star shape, these chicken embryo cells when you grow them in the lab. And then this is the nucleus, and this is the cytoplasm. So you can see these tiny bacteria growing in the cytoplasm. And then when they go into the nucleus, they form these masses. So this image is very characteristic of an infection of cells by Rickettsia. Another two members of alpha Proteobacteria, we have Calyobacter and Hyphomicrobium. They found typically in lakes, and they form stalks and prosticate. And they reproduce by budding rather than binary fission. So here we have Calyobacter. It's a coma-shaped bacteria, and it has this prosticate. So as it's dividing, these flagella will be lost, and then they start forming this budding form, which is a stalk, and then elongates, and then they divide. So this is a film with Calyobacter. And then here you have Hyphomicrobium. So it's budding, and then it has this long extension that is called a hyphae. So this is similar to the extensions of fungi, of mold. So this is a hyphae. Two important bacteria in agriculture, members of alpha Proteobacteria. We have Rhizobium and Bradyrhizobium. They live in symbiosis with the roots of leguminous plants. And then as they grow inside these roots or the rootlets, they fix nitrogen. They obtain nutrients and protection by these rootlets, but they fix nitrogen for the plant, and the plant will grow much better. Another member of alpha Proteobacteria, but in this case it's a pathogen, we have Agrobacterium that causes crown gall. You already saw that. This one inserts a plasmid into the plant cells, inducing a tumor. How is it going to induce the tumor? Well, this plasmid can insert its DNA into this plant cell DNA, and it can prevent the plant from stopping cell cycle division. So it undergoes several rounds of mitosis and causing a tumor. So this is Agrobacterium tumefaciensis causing this crown gall, and then the plant will be short. The tree will be short. In this case, it's a rose plant. Bartonella. Bartonella, we have this specific species, Henselae, that causes cat scratch disease or cat scratch fever. This is a human pathogen. In this world, we have dog lovers, which is equivalent to cat haters, and cat lovers. Cats, guys, not all cats have Bartonella. And also another pathogen, which is Toxoplasma gondii. Only the cats that has this will have uh, Bartonella. Henselae can cause cat scratch fever. But other than that, I'm not giving you more reasons to hate cats if you don't if you don't like cats. But I know that cat lovers, they don't hate dogs. I never have heard anyone like, I hate dogs. But I know that whenever someone don't like cats, they definitely say, I hate cats. And I'm like, okay. Anyway, now for Brucella, Brucella, it's an obligate parasite of mammals and survives inside macrophages. We have a species, Brucella melitensis, that causes brucellosis. So Brucella melitensis can infect cattle, but more than likely will be goats as well. And then some people, they like to drink goat milk or cow milk that is unpasteurized, and they don't even boil it. So then if that animal has this infection with Brucella, it can transmit that into the humans by the milk. And then the person will have something that is called undulant fever. So since this can cause chronic infection, and then that infection can go throughout the life of a person, the person will have fever and then remission, fever, remission, fever, remission, at specific intervals of time. So that's why it's called undulant, because it causes like a wave. And then more members of alpha proteobacteria, we have Nitrobacter and Nitrosomonas. They are part of the cycle of nitrogen. They are chemoautotrophic, so they use inorganic molecules as a source of energy, like CO2 as a carbon source. So Nitrosomonas converts NH4 into NO2, and then Nitrobacter, NO2 into NO3. Wolbachia, it is an endosymbiont in insects and affects the reproduction of insects. So actually, infections by Wolbachia are in Drosophila. It is known to actually determine the sex of the fruit fly. And then this Wolbachia can be used for insect control. They didn't have the image of your textbook. I'm pretty sure that it has the image of an embryo, fruit fly embryo, infected with Wolbachia. They actually immunofluoresce this Wolbachia, so you can see like tiny orange and green dots, meaning that the embryo is infected with Wolbachia. And then with the chemistry. Okay, now let's look into another group of members of proteobacteria. And then which is beta proteobacteria. Before I move on, proteobacteria took its name from the Greek god Proteus. Proteus, it was a Greek god that can take many shapes. So that's why they call these proteobacteria, because as you will be looking at, these bacteria, they have many shapes. So you remember that one that has a hyphae and a bud, those ones that look like a comet shape, those ones they just like plainly rot. So These bacteria can have many shapes. That's why it's called proteobacteria. Now, for members of beta proteobacteria, we have Spirillum, that it can be found in fresh water and moves by a polar flagella. It's a spiral-shaped bacteria. It is shorter and thicker than spirochetes, and it has a single flagella in one end, and it lives in ponds. As Ferrotilus, we have this bacteria that lives in water, fresh water, and sewage water, and it forms sheets. It can be found inside the sheets and reproduced inside the sheets, and it helps them to be protected and to gather nutrients. So here we have this Spirillum volutens. Sorry, I said that it had only on one end the flagella. It has it in both ends, so this is Amphitrachus and polar because it's, of course, at one end. And then this is Aspergillus natans, so this is the sheet. Here is an empty sheet, and here we have the bacterial cells. Here they are duplicating by binary fission. And it duplicates until it fills this sheet. This sheet provides protection and helps for the bacteria to gather nutrients. More members of beta proteobacteria, we can have Burkholderia, Cepacia, and Pseudomyli. These bacteria are medically important. For instance, Pseudomyli causes melioidosis, which is an infection in the human body. And this one, Cepacia, degrades more than 100 organic molecules. Sometimes these ones can be resistant to different antibiotics. And then we have Bordetella, that it is a rod and it's non-motile. We have an important species, Pertussis, that causes whooping cough. We have vaccination against this bacteria. And then someone can die of whooping cough if it's not immune to this bacteria. Neisseria is part of beta proteobacteria. We have two important species, gonorrhea and meningitis. Gonorrhea causes this STD, gonorrhea, that basically these bacteria can infect neutrophils. And then meningitis causes meningococcal meningitis. So zooglia, it is important in the activity of the activated sludge system in sewage treatment plants. So they add these bacteria to start breaking down inorganic molecules within this sludge. So here we have this Neisseria gonorrhea. It's a diplococcus. It is being endocytosed by a host cell. Okay, now let's talk about gamma proteobacteria. We have bacteria like Acidothiobacillus, which is a bacillus that can oxidize hydrogen sulfide into elemental sulfur or into sulfate. So this bacteria is important then for the cycle of sulfur. And then we have Begiota, which grows in aquatic sediments and oxidizes this hydrogen sulfide into elemental sulfur for energy. So these two are important then to recycle sulfur. And then we have Francisella. Francisella tularensis can infect rabbits, and then rabbits can bite a human, causing tularemia. So it's a zoonotic disease. Then we have the Pseudomonadales, in which we have Pseudomonas. Pseudomonas are metabolically diverse. They are found in soil and other natural environments. They are opportunistic pathogens for humans, and they are feared in hospitals. They cause nosocomial infections. They are rods with a polar flagella, and it causes wound infections and UTIs. Pseudomonas aeruginosa. It is highly resistant to disinfectants and also to antibiotics. So here we have a transmission electron micrograph with Pseudomonas, and they are rod-shaped, gram-negatives, of course, with this polar flagella. This is undergoing binary fission, as you can see, by the formation of a septum in between these two cells. When someone has an infection with Pseudomonas, they typically isolate the patient. And then someone goes into the room in the hospital, they have to wear a lot of PPE. They have to destroy that PPE. And then typically, more than likely, it's a wound infection. The person, when you go into that room, has a characteristic smell, the room. It smells like rotten apples. It's very, very distinct, that smell from Pseudomonas. More members of gamma proteobacteria, Azotobacter and Azotomonas, that are nitrogen-fixing bacteria. We have Moraxella lacunata, specifically, that causes conjunctivitis, or infection of your eyes, the external membrane of your eyes, and Acinetobacter, that also it's a fear bacteria in the hospitals. Acinetobacter baumannii causes respiratory infections, pneumonias. Typically can infect these ventilators if it's not, or contaminate these ventilators if they're not properly disinfected. And this one causes a lot of infections, and it's resistant to many antibiotics. From the Legionnalis phyla of the gamma proteobacteria, we have Legionella. Legionella, it is found in streams, water pipes, and cooling towers. There was this convention, I think it was in Philadelphia, from a legion, and they went into this hotel. They had the convention there. They went home, and all the members of the legion developed lung infections. And they isolated these bacteria, and they tried to find what was the source. The source was the cooling tower. It was misting some droplets of water, and then the persons inhale, of course, the bacteria, and they develop this. That's why they call this infection legionellosis. From Legionella pneumophila was the species. And then we have another member of these Legionnalis, Coxiella burnetii. This one causes Q fever, that it is transmitted by aerosols or the ingestion of unpasteurized milk. So here we have Coxiella burnetii. So it forms endospores that are called endospore-like body. And this is making these bacteria to be resistant to heat and drying, just like as any endospores. This is the agent that causes Q fever. We have Vibrionalis, that they live in aquatic environments. We have Vibrio cholera, that causes cholera, which is a severe diarrhea that in some cases can leave the person severely dehydrated, and the person can even die. And we have Vibrio parahemolyticus, that causes gastroenteritis. So here we have this Vibrio, so it's a coma-shaped bacteria, and then it has this polar flagella. Enterobacterialis, part of these gamma proteobacteria. They're commonly known as enterics. They live in the intestines of animals, humans. As a common metabolic reaction, they can ferment carbohydrates, lactose, glucose, for instance. They can live in anaerobiosis. I mean, the intestines, for the most part, they are anaerobic. And they have heterotropous flagella. So they have flagella everywhere. Examples of Enterobacterialis, E. coli. If you find E. coli in food, no matter if it's this E. coli that it's non-pathogenic, like it's part of the normal strain, or if you find it in water, it is a sign of a contaminant indicator from either humans, for the most part, of course, but also from animals. So it's indicator of fecal contamination. We have specific strains that can cause disease, like EHEC, enterohemorrhagic, E. coli, that can cause bloody diarrhea. This is actually the #1 cause of diarrhea worldwide. We have E. coli 015787, and this can cause foodborne diseases and UTIs. E. coli, if I'm not mistaken, the second cause of UTI, the first one is Proteus vulgaris. We have Salmonella with 2,500 serobars or subspecies. This one is as well a common form of foodborne illness. Example, we have for Salmonella, we have Salmonella typhi that causes typhoid fever. That can also contaminate water. More enterobacteria, we have Shigella that causes bacillary dysentery, which is as well like a kind of a very profuse diarrhea with a lot of gas. And then we have Klebsiella that causes Klebsiella pneumoniae. Klebsiella pneumoniae that causes pneumonia. This type of infection by Klebsiella, it is frequently found in people who are homeless and also in alcoholics. They pass out and then they swallow their own saliva. Actually, Klebsiella is part of our normal intestinal bacteria. And then when they basically ingest their own vomit, they can carry part of these bacteria into their lungs and then they can have pneumonia. Serratia, we have Serratia marsensis that produces a red pigment, and it is a common cause of nosocomial infection. Proteus, we have a species, Proteus vulgaris, that causes these UTIs, the #1 cause. And when you put a single bacteria in the center of a Petri plate, can cause a growth that looks like swarming colonies, looks like dunes. So form concentric rings. Yersinia, we have Yersinia pestis that causes plague, and it is transmitted by fleas. And then we have Erwinia that causes infections in plants. So here is Proteus mirabilis. It's a Peritrichus flagella, a rod. See, it can have thousands of flagella, and then if you put one in the center, it will cause these swarming colonies, concentric rings. This is a top view of this growth. Okay, so Enterobacter cloacae and Enterobacter aerogenes causes UTI and nosocomial infections. Chronobacter sakazaki. It is found in the environment, in foods, can cause sepsis and meningitis in infants. Outbreaks are associated with contaminated infant formula. I think we can leave it there. We covered the Enterobacteria. Now, next time we will cover the rest of the gamma protein.